So, imagine for a second that every single cell in your body is like this incredibly exclusive nightclub.
Oh, I love that analogy, actually.
Right. I mean, it has a velvet rope, a highly restricted guest list, and most importantly, it has bouncers working the doors.
Exactly. Because, a tumor just can't grow if it can't eat.
Right.
And for a long time, oncology focused on attacking the tumor itself. But now we are looking at the logistics. We're really looking at the supply lines.
And that's exactly what we're getting into today. Welcome to our deep dive. I'm your host, and today we're exploring a microscopic logistical war alongside our resident oncology expert.
Thanks for having me. I'm really excited to dig into this one.
Same here. So our mission today is to pull from some cutting-edge oncology research, specifically a dense, highly detailed 2026 academic paper from NPJ Precision Oncology.
Yeah, published in partnership with the Hormel Institute. It's a fantastic piece of literature.
It really is. And the whole target of this research is the Solute Carrier transporter family, or SLCs. We want to understand exactly how cancer cells hijack this cellular delivery network.
Right, and how cutting off these specific supply lines could literally be the future of precision metabolic warfare.
It's such a fascinating shift in perspective. Like, this paper basically gives us a blueprint of how a tumor turns a healthy, regulated transport system into this chaotic, greedy supply chain.
Yeah, we aren't just looking at what these transporters do. We're looking at why their specific mechanisms matter so much for a tumor's survival, down to their physical three-dimensional shapes.
So let's start with the architecture of these gatekeepers. When you hear Solute Carrier family, I don't know, it sounds like a small specialized team.
But it's really not.
Not at all. The sheer scale of this is massive. The paper outlines over 450 different proteins broken down into 65 distinct subfamilies.
It is an absolutely massive family. I mean, these are integral membrane proteins.
Meaning they're embedded in the cell.
Exactly. They are structurally embedded directly into the plasma membrane of the cell, as well as the membranes of internal organelles, like the lysosomes or mitochondria.
Wow, okay.
Yeah, and they facilitate the bidirectional transport of basically everything your cells need to maintain homeostasis. Carbohydrates, amino acids, lipids, metabolites.
So they move the raw materials in and pump the metabolic waste out.
Spot on.
And they aren't just passive holes in a wall, right? They're like dynamic machines.
Let's unpack the visual memory tool here, because the structural biology in this paper is wild.
It really is.
It describes how some of these SLCs have what they call left or right-handed G-quadruplex topologies. Which sounds intense.
It does sound incredibly dense, I know. But think of a G-quadruplex as a very specific three-dimensional knot woven right into the structure of the protein.
Okay, a knot.
Right. And the orientation of that knot, like whether it twists left or right, determines what can physically interact with it.
Oh, wow. Yeah, the paper notes that certain small molecules, like one called NMM, will bind exclusively to the right-handed subunits of these knots.
So it's very specific.
It's mathematically precise. One drug molecule binds to exactly one subunit of the knot. There are no loose ends at all. It's a perfect lock and key mechanism.
Okay, so they have these highly complex structures, but they aren't just floating around aimlessly in the lipid bilayer, right? The text mentions they are physically tethered to the cytoskeleton.
Yes, and their function depends heavily on something called MRCs.
Which are what exactly?
Mitochondrial endoplasmic reticulum contact sites. These are the physical structural bridges between those specific organelles.
Okay, structural bridges.
Right, and the paper highlights a vital structural plasticity here. The functional integrity of certain SLC transporters relies entirely on these bridges being intact.
So if the internal architecture of the cell collapses?
The transporters just stop working. In fact, the paper notes that pharmacological agents like melatonin can actually restore MRC architecture.
Which in turn enhances the transporter function. That is fascinating. The transporters are intimately tied to the structural scaffolding of the cell.
Exactly.
Let's do a quick recap here. So we've got 450 of these transporters, these complex 3D structures that are super dynamic. And I want to ask you, the listener, a question.
That's a good one to think about.
Yeah, think about your own body right now. How many of these millions of microscopic gates are opening and closing just while you listen to this? It's mind-blowing.
It really puts it into perspective.
It does. But here is my question. If these SLCs are vital for basic biology, like recycling neurotransmitters in the brain or excreting drugs through the kidneys, how does cancer specifically hijack them without just instantly killing the host cell?
Well, because cancer doesn't break the system. It hyperactivates it.
Okay. Oh.
A tumor selectively upregulates, meaning it transcribes and translates way more of the specific transporters it needs for rapid growth.
And leaves the rest alone.
Exactly. It leaves the rest of the network totally alone. It's a targeted, hostile takeover of the supply chain.
That is terrifying. Let's look at exactly what happens when cancer forces those specific doors wide open. We're moving into the sugar-acid cycle now, which brings us to the Warburg effect.
Yes. The Warburg effect is, it's one of the foundational hallmarks of cancer metabolism.
The sugar greed, basically.
Yeah, rapidly proliferating tumor cells are incredibly greedy for glucose. Even when oxygen is perfectly available to run normal, highly efficient aerobic respiration, cancer cells will gorge on glucose to run glycolysis instead.
Which is weird, right? Because glycolysis is less efficient.
It's much less efficient for generating APP, yes, but it is incredibly fast. The cancer cell prioritizes speed over efficiency.
And to consume that much sugar, they physically need more doors.
Yeah.
Specifically, the paper points to GLUT1 and GLUT3, which are part of the SLC2 family. But how does a tumor force the cell to build more of these glucose stores?
It relies on a mechanistic cascade driven by stress. As a tumor grows, it creates a chaotic mass of cells that often outstrips its own blood supply.
Creating a low oxygen environment.
Exactly, a highly hypoxic environment deep inside the tumor. And under hypoxia, a transcription factor called HIF-1 alpha stabilizes and accumulates.
Okay.
HIF-1 alpha physically migrates into the nucleus, binds to the DNA, and directly upregulates the genes responsible for manufacturing GLUT1 and GLUT3.
But wait, if HIF-1 alpha is building more doors specifically because oxygen is low?
What happens when it's not?
Right. What happens when the cancer is just growing aggressively regardless of oxygen levels? How does it keep the supply lines open when it isn't suffocating?
That is where oncogenes take over, specifically c-Myc. The paper highlights that in cancers like lymphoma, c-Myc is wildly overexpressed.
So it doesn't need a hypoxia trigger.
Exactly. c-Myc binds directly to specific DNA sequences called E-box elements on the GLUT1 promoter. This just supercharges the transcription of the transporter. And what's brilliant, well, from a survival standpoint, is that c-Myc simultaneously upregulates the downstream enzymes needed to process that sugar.
Like hexakinase 2.
Yes. So it builds the doors to bring the sugar in, and it builds the processing plants inside to burn it.
But there's a massive biological consequence to burning sugar that fast through glycolysis, isn't there? You generate a lethal amount of acidic waste.
Yes, specifically lactate.
Right. And if lactate builds up inside the cell, the intracellular pH drops, proteins denature, and the cell literally chokes on its own exhaust.
Yeah.
How does it prevent itself from dying in that acidic environment?
It basically installs a heavy-duty garbage chute. Specifically, a transporter called MCT4, which is part of the SLC16 family.
Okay, so MCT4 is a proton-coupled transporter, right?
Yeah, it takes the lactate molecule, pairs it with a proton, and violently pumps it out of the cell.
So the internal pH remains stable, the cancer cell survives, but now the outside environment, the tumor microenvironment, becomes highly acidic.
It does. And if we connect this to the bigger picture of oncology, that acidic moat outside the cell is a masterstroke of immune evasion.
Because it paralyzes our immune cells.
Exactly. Our immune cells, particularly CD8 plus T cells, whose exact job is to hunt and kill tumors, become paralyzed in highly acidic environments. Their metabolic machinery just shuts down.
Wow. So by pumping out its own acidic trash through MCT4, the tumor is inadvertently building an invisible chemical shield that repels your immune system.
That's incredibly effective.
That is terrifyingly elegant. And it brings up a specific case in the paper that genuinely shocked me regarding insulin.
Ah, yes, the hyperinsulinemia connection.
Yeah. So we know the hormone insulin lowers blood sugar by pushing a different transporter, GLUT4, to the cell membrane to pull glucose out of the blood. Does this mean a patient with hyperinsulinemia, like in type 2 diabetes, is inadvertently providing their cancer cells with more doors to feed through?
Unfortunately, yes. The source text confirms this exact mechanistic link. In breast and prostate cancers, dysregulated insulin signaling through the IGF-1R pathway facilitates the translocation of GLUT4.
Okay, break that down for us.
Sure, normally GLUT4 is stored in vesicles deep inside the cell, but constant high insulin levels trigger a signaling cascade that forces those vesicles to traffic to the plasma membrane and fuse with it.
So hyperinsulinemia is essentially handing the tumor's bouncers VIP passes, leaving the glucose doors permanently propped open.
That's a great way to phrase it, yes.
Okay, let's do another quick recap. GLUT1 brings sugar in via HIF-1 alpha and C-MyNE. And MCT4 pumps acid out to build an immune shield.
Right.
So listener, if you were a cancer doctor, would you try to block the food coming in or block the trash going out? It's a tough call.
Both are valid targets, absolutely. But sugar is only one part of the equation.
Right, because to truly understand the tumor's logistics, we have to look at how it physically builds itself and defends itself. We need to talk about amino acids.
The building blocks. The paper lays out several amino acid transporters, but I think the best way to visualize this network is as 3 distinct interconnected pillars.
The 3-pillar concept works perfectly here. Let's start with the first pillar, the builder. This is LAT1, right? Scientifically known as SLC7A5.
Exactly. LAT1 primarily imports large neutral amino acids with a heavy preference for leucine.
And leucine isn't just raw material. It acts as a biological trigger. When LAT1 pulls leucine into the cell, it flips a major metabolic switch called the mTORC1 pathway.
Spot on. mTORC1 is essentially the cellular foreman. When it senses high levels of leucine, it signals that raw materials are abundant.
Which drives massive protein synthesis.
Yes, protein synthesis, cellular proliferation, and adaptation to stress. High LAT1 expression means that growth signal is permanently jammed in the on position.
Okay, so LAT1 builds the physical structure, but rapid, unchecked growth requires massive amounts of heavy-duty energy. Where's the fuel coming from if glycolysis is so inefficient?
That brings us to pillar 2, the engine fuel.
Which is driven by ASCT2 or SLC1A5.
Right. This transporter's primary job is to aggressively pull in glutamine. Once inside the mitochondrial matrix, glutamine undergoes glutaminolysis.
Okay, converting into what?
It eventually converts into alpha-ketoglutarate. That molecule is fed directly into the TCA cycle, the Krebs cycle, which is the cell's primary heavy-duty energy engine.
So it provides the massive ATP power needed to sustain rapid cellular division that glycolysis alone just can't support.
Exactly. It's the heavy-lifting energy source.
And finally, we have pillar 3, the shield.
Yes. The shield is governed by XCT or SLC7A11. Mechanistically, this one operates as an antiporter, an exchanger.
Meaning it swaps things?
Right. It pumps one molecule of glutamate out of the cell in exchange for pulling one molecule of cysteine in.
And why is cysteine so important?
Because once inside, cysteine is rapidly reduced to cysteine, which is the rate-limiting precursor for manufacturing glutathione or GSH.
Which is the master antioxidant of the cell, right?
Exactly. Rapidly dividing cancer cells run their metabolic engines so hot that they produce massive amounts of reactive oxygen species.
Essentially biochemical sparks that shred cellular structures.
Yeah. If a cell takes too much oxidative damage, the lipids in its plasma membrane undergo lipid peroxidation. The membrane literally shreds itself in a specific type of cell death called ferroptosis.
It's essentially the cell rusting to death.
Precisely. So by upregulating XCT, the cancer cell constantly pulls in cysteine, constantly produces glutathione, and neutralizes those sparks.
It prevents ferroptosis and builds a robust antioxidant shield against oxidative stress.
And this is vital because oxidative stress is exactly how many traditional chemotherapies attempt to kill tumors.
Wow. What I find most fascinating is that these pillars aren't acting in isolation. The paper points out that ASCT2 pulling in glutamine actually facilitates LAT1 pulling in leucine.
Yes, they are chemically cooperating. They cross-feed the system.
To ensure the builder, the fuel, and the shield are all operating at maximum capacity.
It really highlights that oncology can't just target one transporter and expect the whole system to collapse. It is a highly integrated compensatory metabolic network.
Time for recap number 3. We've got our 3 pillars. LAT1 pulling leucine for the mTOR builder, ASCT2 pulling glutamine for the TCA engine, and XCT pulling cysteine for the antioxidant shield.
A perfect summary.
Notice how cancer doesn't just steal energy. It steals the exact specific materials needed to build chemical armor against our own immune system. So listener, which of these 3 pillars seems like the most vulnerable target to you?
It's definitely something researchers are debating constantly.
Before we get to how we strike back at these pillars, we need to address the other major raw materials, the paper details. Lipids and nucleotides. These sections are dense, but they reveal so much about chemoresistance.
They absolutely do. Let's look at the FATP family, or SLC27, which imports long-chain fatty acids.
Right. We often think of lipids just as structural components for new cell membranes, right?
We do, but cancer cells utilize FATP transporters to pull in lipids and store them as intracellular lipid droplets.
And why go through the effort of storing them?
Because those lipid droplets act as a literal chemical sink.
A sink, like to trap things.
Exactly. When a patient is bombarded with lipophilic chemotherapy drugs, Those lipid droplets can sequester the drug, trapping it away from the nucleus where it's actually supposed to do its damage.
Well, that's incredibly sneaky.
And furthermore, altering the lipid composition of the plasma membrane changes its fluidity, physically preventing certain drugs from entering the cell. It is structural armor.
And then you have the ENT family, or SLC29, which handles nucleotides.
Right. If a cell is dividing that rapidly, it has to replicate its entire genome every single time.
So the nucleosides pulled in by the ENT transporters are the literal genetic letters, the A, C, T, and Gs, needed to synthesize new DNA and RNA.
Exactly. If a tumor lacks these, the DNA replication forks collapse, causing massive replication stress and ultimately cell death.
So we have a complete picture of the logistics now. The glucose doors for rapid energy, the trash chutes for acid, the amino acid pillars for growth and antioxidant shields, the lipids for structural armor, and the nucleotides for genetic replication.
It's a comprehensive supply chain.
So what does this all mean for patients right now? How do we turn this logistical map into an offensive strategy? What are the actual counterattacks mentioned in this paper?
Well, the pharmacological landscape is moving aggressively toward targeted SLC inhibitors. Let's start with the glucose stores. We have highly selective GLUT1 inhibitors like BAY876.
Okay, what does BAY876 do?
It physically blocks glucose uptake, drastically depleting intracellular ATP production, especially under those hypoxic conditions we discussed.
That makes sense.
Interestingly, it also shows immunomodulatory properties, meaning starving the tumor of glucose might actually alter the microenvironment enough to help immunotherapies work better.
But there's a massive biological hurdle here, isn't there? GLUT1 isn't exclusive to cancer cells.
No, and that is the ultimate bottleneck. GLUT1 is the primary glucose transporter for your red blood cells, and it heavily populates the blood-brain barrier.
Oh, so systemically inhibiting GLUT1 with a drug like BAY876 causes severe off-target toxicity.
Right. You cannot systemically starve a tumor of glucose if you are simultaneously starving the patient's brain.
Which is an incredibly narrow therapeutic window. What about blocking the trash chute, MCT4? If we can't starve them safely, can we drown them in their own waste?
That approach is actually showing immense promise. There is a prominent MCT4 inhibitor called VB124. By blocking the lactate chute, the tumor chokes on its own acidic exhaust.
Nice.
But the downstream effect is even more important. It drastically reduces the acidity of the surrounding microenvironment.
It lifts the acidic moat.
Exactly. And by lifting that moat, it reactivates the paralyzed CD8 plus T cells. Preclinical models in the paper show that using VB124 actually enhances the efficacy of PD-1 immune checkpoint inhibitors.
Wow, so you are removing the chemical shield so the immune system can finally see and attack the target.
Precisely.
What about our amino acid pillars? Can we dismantle the builder, the fuel, and the shield?
We have compounds targeting each. For the builder, LAT1, there is a drug called nanvarenlat, which blocks leucine uptake, effectively turning off that mTOR growth switch.
And for the engine fuel?
For ASDT2, compounds like V9302 cut off the glutamine supply line. But the really fascinating approach is targeting the shield, XCT.
Right, the antioxidant shield. How do we break it?
Using drugs like arrestin or sulfasalazine, we can block the cellular uptake of cysteine. The cancer cell can no longer manufacture glutathione.
And without that antioxidant, the massive amounts of reactive oxygen species it produces shred its own membranes.
Exactly. You are pharmacologically forcing the cancer into ferroptosis. You are forcing it to rust to death.
But we come back to the toxicity problem, don't we? If we systemically block amino acid uptake, we harm healthy, dividing cells across the body. How do we solve the delivery problem?
It's $1 million question. How do we poison the specific supply trucks without destroying the entire highway system?
Exactly. So I'll ask the listener. Imagine trying to poison the exact delivery trucks that your own brain relies on to function. How would you solve the delivery problem?
Well, this is where the paper dives into nanotechnology. Researchers are developing targeted nanoparticles to bypass systemic toxicity.
Okay, nanoparticles. Like what?
For example, the paper highlights a nanoinhibitor complex called ZNPCS-ACO. Instead of flooding the bloodstream, this nanoparticle is engineered to target SLC38A9 directly on the membrane of the lysosome.
Which is essentially the stomach of the cell.
Right. It uses the tumor's own acidic microenvironment to trigger the release of the payload exactly where it's needed in minuscule doses. This virtually eliminates off-target toxicity to healthy organs.
That is absolute precision. But part of precision is realizing that not all cancers use the same supply lines. Which brings us to the personalized war.
Yes. The specific SLC targets change drastically based on the tissue of origin, the specific genetic mutations, and even the etiology, what caused the cancer in the first place.
Tumor heterogeneity dictates everything. You have to profile the specific tumor to know which door to lock. The paper breaks down gastric cancer as a perfect example of this.
It does. If a patient has the intestinal subtype of gastric cancer, the tumor relies heavily on LAT1, our builder pillar.
But if it's the diffuse subtype,
then it shifts its reliance to ASCT2, the glutamine fuel pillar. And if it is a HR2 positive gastric tumor, its primary vulnerability is MCT4, the lactate garbage chute.
the same logic applies to liver cancer too, right? Hepatocellular carcinoma.
Exactly. The etiology changes the metabolism. If the liver cancer was induced by a viral infection like hepatitis B or C, the tumor typically relies on GLUT1 for massive glucose uptake.
But if that exact same type of liver cancer was caused by non-alcoholic fatty liver disease, it rewires its metabolic dependency toward lipid transporters like FATP1.
It's incredible how adaptive it is. And in lung cancers, it comes down to the specific genetic mutation. Non-small cell lung cancers with an EGFR mutation rely heavily on ASCT2 for glutamine.
But if that lung cancer is driven by a KRAS mutation, it becomes hyperglycolytic, right? It completely depends on GLUT1.
Right, you have to pair the specific SLC inhibitor with the specific genetic profile to get a synergistic effect. It requires mapping the dependencies of the individual tumor.
Precision metabolic intervention is definitely not a blunt instrument.
Not at all.
I do want to push back on one thing, though, because the paper brought up a fascinating paradox with breast cancer regarding a transporter called OATP, or SLC01.
Oh, the tamoxifen paradox, yeah.
Yeah. We spent this entire time talking about how to block these transporters. But it turns out high expression of OATP actually facilitates the cellular uptake of the chemotherapy drug tamoxifen.
It does.
Wait, so sometimes we actually don't want to block the transporter. Sometimes the transporter being highly active is actually the exact mechanism our chemotherapy uses to get inside the cell.
That is a brilliant observation, and the paper explicitly confirms this paradox. In estrogen receptor-positive breast cancer, clinical data shows that reduced expression of OATP is directly linked to resistance to endocrine therapy.
Wow, the cancer gets smart. It realizes the drug is using a specific door, so it simply removes the door.
Exactly. In that specific scenario, the therapeutic goal flips completely. It is no longer about blocking the transporter. It's about finding ways to restore or enhance its function. Sometimes you really need to hold the door open to deliver the poison.
So, what does this all mean for you listening? It means the future of cancer treatment isn't just carpet bombing the entire body with chemotherapy and hoping the tumor dies before the healthy tissue does.
No, it is precision metabolic warfare.
It's about reading a tumor's specific SLC signature, finding out exactly which bouncers have been corrupted, and starving the tumor to death by closing its exact logistical doors. Or, in the case of tamoxifen, exploiting the exact doors it foolishly left open.
And this raises an important question, something the field of oncology is just beginning to explore.
What's that?
Well, if cancer's greatest survival trick is upregulating these specific SLC transporters to greedily pull in nutrients, and we know that certain transporters pull in specific molecular structures,
Right.
What if the future of therapy isn't just about blocking these gates, but actively using the cancer's own greed against it? What if we design Trojan horse drugs that perfectly mimic the exact amino acids these hyperactive SLCs are desperate to consume?
Like hiding a lethal chemical payload inside a nutrient disguise. Using the nightclub bouncer's own VIP list to sneak in the ultimate undercover agent. That is an incredible thought to leave on.
It's an exciting frontier.
Next time you think about your biology, remember those 450 microscopic gatekeepers working tirelessly in the background, determining exactly what gets in and what stays out. Thank you for joining us on this deep dive. Keep asking questions and keep exploring.